Alright, on to our third video, which is going to be relatively short in comparison to the others. We're going to basically talk about buffering systems, meaning we're really going to get into what pH is, how pH is calculated, and then kind of why it's important for us to maintain a relatively normal or tight range of pH, and how do we maintain that pH without causing wild fluctuations. And we'll also get into one last thing, alkalosis and acidosis, where we have lost control of that pH level and it has swung outside of the normal range. So before we get into talking about all this, we have to actually define what pH is. But pH is basically just the measure of the hydrogen ion, also known as the proton. So we just want to know how concentrated is the hydrogen ion in this solution. Now it runs from 0 to 14, 0 being the strongest acid, meaning the highest concentration of those protons, and 14 being the strongest base, meaning it has the lowest concentration of those hydrogen ions. So if we look at how it's calculated, pH is just the log of 1 over the hydrogen concentration. So if we look at the scale, right, we see like 10, we see our 1 through 14, but we notice all those exponents are negative. And if you remember from our lab from week 1, all those, if the negative exponent is small, like -1, it's actually bigger than, say, -4, right? It's going to be a bigger number. So a higher concentration is going to be at the lower pH. That's why we see it. It's a reverse logarithmic scale. Now, pure water has a hydrogen concentration of 10 to the -7, so it is neutral, right in the middle. Now, water is great at being a solvent. It's really good at resisting changes in temperature, but it's not good at resisting changes in pH, right? And so we need other things to help, namely what we call buffers, to help prevent wild fluctuations in pH. When we look at our own body fluids, homeostasis is maintained at a pH of about 7.4, roughly, give or take a little bit. And so we need buffers to stabilize this pH so that we can stay around the 7.4 and not have wild changes. So buffers resist this basically by taking those strong acids or bases and converting them into weaker acids and bases. One of the most common ones we see in the body is bicarbonate, or HCO3 negative. We can add a hydrogen ion to this so we can pull up the excess of those protons to form carbonic acid, but we can very easily go right back and forth. So we see like this bicarbonate all over the place, especially in the blood, to make sure that the blood pH does not fluctuate wildly, that it stays relatively the same. Now, we said that there's a very narrow range. So about 7.4 is considered a normal pH of the blood. If we go down too low, 7.35, right, we get lower than that, we say that the person has acidosis, meaning their blood is too acidic. If it goes over 7.45, the patient has alkalosis, right, too basic. Both of these can be in our life-threatening, right? So think about that, 7.4. So we have a 0.05, right, give or take, right? That's all the room we have. That's the room for error we have before we start to feel sick and or face severe health consequences and or die.